Okay. Um, Today I'm going to be presenting a project that I did for my third rotation, which was in a microscopy lab. So my project was basically to optimize this uh, cargo transport assay, and so the purpose was to like visualize and measure cargo transport to the Golgi. And here's a little layout for you guys so you can orientate yourself throughout the presentation. Okay, so before I can ask a question, we have to know what a Golgi and cargo is. So the Golgi regulates the transport and modifications of proteins and lipids, usually by glycosylation. Um, and it takes, let me get my pointer. The ER sort of transports proteins before they can get modified, and so they leave ER exit sites and travel down microtubules to the Golgi, and they move from positive to minus ends of the microtubule. So that's kind of interesting since like the rest of the cell usually uses minus end to positive end. So this is like a dynein dependent transport. And I think the next page here, I have a better diagram of like the actual Golgi. So you get stuff from the ER, and I kind of try and visualize it like a lava lamp or something similar. So once it gets there, this is like the end of or the start of the microtubule, and you get protein going into the first stack or cisterna of the Golgi. These things are separate, so the most common example I've seen is you think of pancakes, like a stack of pancakes. And so you have protein going from one cisterna to the next, but you also have this sort of constant change. So you also have lipid compositions transferring down. So this transmembrane cisterna, or transgolgi cisterna, will exit out into the cell for, like, it's going to hold all these proteins that's ready to go for the cell, and then so as that one's leaving, it's then kind of turning the cisterna below it to be trans. So these have different lipid compositions, and so it's important for them to transfer. That's just like kind of background that doesn't really have anything to do with my rotation project, but I thought it was pretty interesting. This is sort of I want to go through an overview of kind of the actors in the play. So we have our Golgi, which we just talked about, and then we're in like the average sort of cell, right? G1 cell phase, epithelial cell, right? That's, I think, what I worked with. You have the Golgi co-localizing with the centrosomes. So there's actually two centrosomes here. And so usually when you see the Golgi, you know that's where the centrosome is, and vice versa. And then we have this sort of purpley line as the ER, and then the circles as ER exit sites. This diagram I made just to try and like visualize things, but it's not actually like known to work like this. So like we don't know how the microtubules sort of we don't know like the orientation of them, so we don't know if they like go together or they're completely separate, like individual lines or whatever. So that's an open question. And yeah, I mentioned dynein. It's just a dynein dependent transport. Why you should care. So any sort of fundamental cell process is going to impact like everything. So you could come up with any number of diseases or disorders that have dysregulation in protein transport, or we'll see hopefully later that the Golgi will change its shape, and without that the cell cannot divide. And so that's also important, right? If the cell can't divide, then that has translational aspects, right? Okay. Also, as a researcher, you might want to care because there's a lot of open-ended open questions about the Golgi and how this whole process works. And so, okay, now we can get into the actual project, where my question is, how does the Golgi morphology affect cargo trafficking? And to give some background on like why that matters, why we're asking that question, right? Depending on the cell cycle, you have either a compact Golgi or an extended Golgi. And this is something that's very specific to the Kaverina lab that I rotated in. They like discovered this, and it's like no one knows why, so that's kind of exciting. And so like I want to measure if cargo transport, I should have mentioned cargo just means like proteins in like vesicles going down the microtubule. And so I want to measure if the protein transport to the Golgi differs between this compact state versus this extended state. And yeah, this is a little diagram that kind of shows how the Golgi changes throughout the cell cycle. I don't particularly like this, but there wasn't really a better diagram. So like I said, the hypothesis that we're testing is that the rate of cargo trafficking varies between the compact versus the extended Golgi morphology. And the rationale behind this is just like the distance between the ER exit sites is changing between morphologies, and also so this is, I'll follow along, I'll show you as this GIF progresses. Okay, so this is G1, this is normal cell state, then we're going to S phase where you're replicating everything, and then this is right before mitosis, and then this is in mitosis, and or I think prophase, it's prophase. And so as the cell goes through preparing for mitosis, there's a few things that the cell has to sort of organize beforehand. So the nucleus, the envelope, will disintegrate, and I tried to show it here. So this is the the outside of the nuclear envelope. It's a bilipid membrane, and the outside layer is continuous with the ER, and so the ER sort of takes in a bunch of the nuclear envelope proteins, and so like nuclear pore things and like transmembrane proteins and like things that like hold the nucleus in place. And so that will all go into the ER. And so another rationale is you're having different compositions in different stages, and so that might affect trafficking somehow. And then surface area of the Golgi, that's also a thing. And then also something really cool is that when the Golgi starts to do this extension, it actually becomes centrosome independent. And so how I said before that the Golgi co-localizes with the centrosomes, that doesn't happen. Like it disassociates. And I think that's really interesting because where the centrosomes are and like where the Golgi is normally is like the microtubule organizing center. And so it's like really like you can think of like a railroad map or something like that where like all the lines converge. That's where the centrosomes are. And so if that's changing, if its dependence on that is changing, I would think that the trafficking also changes. I don't know how to change it, so I'm sorry, but there's this sort of dimer, what is it called? I'm sorry. FKBP, and so here you have the tetramer, which is the DS red, and then on the ends you have this FKBP, and so you have this sort of aggregate form of the protein, so it's like it's all bunched together, and then when you add in SLF, which is a synthetic FKBP, so it's like just the dimer part, and so the SLF will dimerize with the FKBP, and so that allows the proteins to be sort of separate from each other and not all clustered. And so that's important for the release of the protein. So like before you add SLF, all the proteins cannot get transported out of the ER. They're just too big. And after you add SLF, they then are small enough to get transferred. And so my experimental steps, I haven't really worked with wet lab that much, and so it was pretty fun to learn how to like seed cells and things. So, and this was the actual like transfection. So we used a transmembrane trans-Golgi marker, and also the ES cargo marker, and we also used Halo as a marker for ER exit sites, but those aren't as important to answering the hypothesis as the other two. And yeah, I learned how to like basically take care of cells, and before you can make a movie and like at SLF and see the transfer happening, you need to find a good image. And so you look through the microscope and you try and find a cell that is successfully transfected with both the Golgi and the cargo, or in this right picture, the Golgi, the ER, the cargo, and the ER exit sites. So I should have put it, but you should have labels for all of these things. I forgot to add it. That was one of the biggest comments that I got on this presentation when I gave it to the lab. So yeah, going forward, Golgi will be green. Cargo will be red or magenta, and then ER exit sites will be the cyan color. You see all the cargo kind of disperse before going to the ER, and so I was working with a postdoc, Avi, and we both didn't know how to do it, and so that was kind of like my project, was to learn alongside with him. And so one of the main limitations of the assay was that they say it in the paper that the ES cargo came from, that in mammalian cells sometimes it's hard for the cargo to leave the Golgi. So it can go to the Golgi, but it can't leave it all the time. Sometimes it happens, sometimes it doesn't. Also, yeah, it gives, so this is basically the wild type or something, right? You're trying to get a baseline or a benchmark to do analysis, and so you need to know how it normally happens to then try and synchronize the cells to be in different cell cycle phases. And so that's kind of the end goal here, but my rotation wasn't long enough to do that. And yeah, if we could try and fix the cargo moving out of the Golgi, we can measure that rate too. So we're trying to measure the rate of cargo to the Golgi, but in the future maybe you can measure cargo from the Golgi out. Okay, so my original video got messed up with the edits because I went between slides. Okay, so both of these videos or movies are when the SLF didn't work. And so again, cargo is in magenta, Golgi is green. Excuse me. And so yeah, I think this one was done in the spinning disc confocal, and I think this was the end spark. And in this one, this is end spark for sure. It also didn't work, but you can see there's a lot of detail there. Also, something cool to note is even though the SLF didn't work, you still see some type of cargo dynamics. So you see this directional movement of the cargo on some areas, and then in others it's pretty much stationary. And so that's kind of weird, right? We don't know why that's going on. So it's just an interesting observation. In these two movies, we have the SLF actually working, And so you see cargo getting transferred to the Golgi, staying there, and then getting released. And in... oh, I don't have my pointer on, I'm sorry. In this video, you can actually see the cargo leaving, like right there. So you can... it's pretty cool or interesting to see the dynamics of the cargo, and yeah, I'm pretty proud of these for someone who has never touched a microscope before. Well, I have, but not like this. It was really exciting. And I think my next slide is fine. Pretty cool stuff. I think these worked too. Yeah, for this one, so the Golgi's in green here. I think this one doesn't have the Golgi marker, but I thought it was interesting how sort of the ER exit sites. So the marker that we used, the halo thing, doesn't actually mark the ER exit site specifically. It marks a vesicle that is specific to the ER exit site, and so it marks a vesicle that moves the cargo or moves the proteins. And so I thought it was interesting to see sort of that vesicle stay in the Golgi. And so the reason why I thought that was interesting is that probably affects trafficking dynamics. And so if you're not recycling the vesicles, like that's a pretty like important process to get the vesicles that transfer the protein to the Golgi back to the ER exit sites. And so you see this pretty stark delay. Like you don't really see the ER exit sites leave. And I should note that like this is not just one movie, this is in all of the movies, and so it's not just this artifact. So in these movies, we have the ER exit sites also labeled. Just more videos where it worked. And yeah. I mean, I think they're just pretty to watch. I don't know how much you can really get from it. Again, this is purely to get a baseline for future analysis. And so there's not much else to be said about it, but I still think it's cool. The acknowledgements. So Avishkar is my mentor for the lab. He's a postdoc in Irina Kaverina's lab. And Maggie Faye, she is about to graduate, so soon she'll have a comma PhD next to her name. And Henrik was a rotation student that rotated in the lab alongside me. And then I'm covering Saeed and Raul. They're also postdocs. And then Hamida is like the backbone of the lab. She is so important. She's basically the lab manager. And then I have sort of a personal slide that I showed, but you don't have to include this if you're doing a presentation like this, but I wanted to give everyone my email. This is kind of a silly story. So like I went hiking with my family, and to Ringing Rocks, which is in New York, and you can like hit the rocks and it makes noise. But we went there and this was carved into a rock. Like, I didn't do this. It's my name. And so I thought it was like kind of freaky, but I was trying to find an image that would like sort of represent me, and I thought this was okay. And I also mentioned to the lab that I have this YouTube channel. So